Chromatin remodeling. When one thinks of chromatin, it is always considered or it is always associated with a condensed state of DNA. And when you use the word remodeling with chromatin, it basically implies that the chromatin structure is changed or it is rearranged. And this rearrangement of chromatin has implications on several biological functions or molecular mechanisms such as replication, DNA repair, transcription, etc. So let us look at the learning outcomes of this session. The outcomes are packaging of DNA into chromatin not just condenses the DNA, but it also can regulate gene expression. Chromatin remodeling can initiate or repress transcription. Histone modifications, especially of the nucleosome core particles, influence chromatin formation and also influence gene expression. Chromatin remodelers are molecules of about 5 different families that modify and mobilize the nucleosomes by various mechanisms that are all ATP dependent. This is important. Chromatin remodelers can slide nucleosomes, can evict nucleosomes, can disassemble the nucleosomes in such that a dimer is removed or a tetramer is removed, replace canonical histones with histone variants. So now let us look at a little overview or aspects of chromatin remodeling. First, let us understand what do we really understand by chromatin remodeling, or what is basically the need for chromatin remodeling. So let us consider over here this being a bead and structure is bead and string structure. This is basically the DNA wrapping around every nuclear core particle, with the DNA wrapped around being the core DNA and the DNA between two adjacent nucleosomes being the linker DNA. Suppose the linker DNA is also bound by histone, that is H1. This bead in the structure further gets into a higher order structure like the 30 nanometer fiber or the solenoid form or the zigzag form of the chromosome. So that becomes a second level of condensation, this being the first level of condensation. Now, the thing that we observe here is therefore that the DNA is packaged. Okay, so suppose you have a promoter region for a gene present in the linker DNA. If that is the case, the DNA is accessible for the RNA polymerase to come and bind. And once the RNA pol comes and binds to the promoter region of the gene, it will begin transcribing the gene. But it can easily transcribe until the DNA is not wrapped around the nucleosome. But the DNA that is wrapped around the nucleosome is going to be difficult to transcribe because the DNA is already bound to the nucleosome core particle. So the accessibility of this part of the DNA is less for the RNA polymerase. And hence what can happen is that the transcription can stop or slow down or halt. Now let us look at another scenario where you have the promoter region in the core DNA. That is, it lies within the core DNA. In such a case, this promoter being already bound to the protein, and we know that the histone and the DNA form 14 contact points. So therefore, the binding of the DNA around the or wrapping of the DNA around the nucleosome is not loose. It is tight and rigid and it is stable. So therefore, for the RNA polymerase to get access to this promoter region is less or is next to negligible, and so this gene will not get expressed. Therefore, what we are understanding is that the chromatin per se is making a gene inaccessible to the RNA polymerase, and hence transcription will be not taking place from this promoter. Therefore, what one can understand is that if a promoter is present here, it may get access to the RNA polymerase and it may begin transcription. So that gene gets transcribed. But if you have a promoter within the core DNA structure, then the accessibility to RNA poly decreases and therefore transcription will not happen. So in other words, very clear is the fact that the nucleosome is acting as a barrier for transcription. Okay, the same can be applied to replication as well. For example, if the origin of replication is within the chromatin region, then the opening up of the DNA at that origin becomes difficult because you cannot have the initiator coming and binding to basically unwind the DNA to form the two templates. So this is something that is there with relation to any molecular mechanism. For example, you have a DNA within the core DNA, you have a mismatch or you have a DNA lesion. Then again, for the DNA repair mechanism to come and repair it at the region where there is a nucleosome bound is going to be difficult. So therefore, the understanding is very clear. More compact the DNA becomes, less accessible the DNA becomes to all molecular machineries. So transcription will not initiate. And for this to be taken care of, the main thing that has to be done is it will have to loosen the tight wrapping of the DNA around the nucleosome so that the DNA becomes more accessible to the transcription machinery or to the replication machinery or to the DNA repair machinery. But uh, in this entire session, we would be relating this more with transcription um, because there are many examples that have been found with respect to chromatin influencing the transcription of genes, influencing gene expression. Now, therefore, this chromatin basically acts as an epigenetic change or epigenetic thing. So, if you have a chromatin bound to a certain region of the DNA, then it means that DNA is not accessible and therefore gene is not getting expressed. Okay, so that is related to epigenetics. Now, one of the ways by which this wrapping of the DNA around the nucleosomes is affected is by histone modifying enzymes. So, this is one basic family of enzymes or proteins that help in chromatin remodeling. We all know that when you have the nucleosome core particle formed, all the eight histones that are present, H3, H4, H2A and H2B, have their end terminal ends extended out. So these are what are called as the histone tails, and these histone tails are hot spots for modifying enzymes. So they are spots where post-translational modifications can happen. So you could have phosphorylation and serine residues or tyrosine residues. You can have acetylation and the lysine residues. So the acetylation is carried out by enzymes that are called as histone acetyl transferases, in short called as HAT. And you can have enzymes called deacetylase or sorry histone deacetylase, that is HDAC, and either of the two are reversible. So if certain lysine residues have been acetylated by the HAT, you can have the HDAC coming and removing the acetyl group from the same. You can have methylation by methyl transferases. You can have ubiquitination as well. So several different kinds of modifications are possible at the histone tails. And interestingly, depending on the type of modification that is happening, you have very specific patterns of modification. So say for example, this is one pattern of modification. Then this can be one histone code. Say this is another pattern of modification, then this becomes another histone code. This becomes another histone code. Now, these histone codes that are being written by these histone modifying enzymes can be read by other molecular mechanisms, other molecular complexes. And also what has been observed is These modifications, be it acetylation, deacetylation, phosphorylation, or methylation, can either make the chromatin more rigid and so therefore less accessible, or they can make it more open. That means the winding of the DNA around the nucleosome core particle can be loose, and if it is loose, then the DNA can loop out, and if the DNA loops out, then you can have it accessible to other non-histone proteins. So therefore, for example, if you have acetylation, the acetylation can make a positively charged lysine into an uncharged residue if it gets acetylated, because acetyl group is COCH3 and COCH3 does not have a charge on it. But if you did not have a COCH3, the lysine will have a positive charge. So if the lysine has a positive charge, its affinity for binding to the DNA is greater. But if it is acetylated, its affinity to bind to the DNA is going to be less. Hence, acetylated histone tails is an indication that the DNA can be transcriptionally active. But a deacetylated histone tail is an indication that it can be transcriptionally repressed. Okay, so this is an interesting and important phenomenon. And like mentioned, these histone codes that are formed, they can be read by different complexes. And these molecular complexes are, say, for example, molecular chaperones. So we all know that molecular chaperones are complexes that can help in folding or unfolding of DNA, or can help in assembling or disassembly of proteins. So therefore, they can come and bind to the DNA through these histones, or you can have chromatin remodelers which can come and bind to the nuclear core particles. So histone tails are basically binding sites for different molecular complexes, and the binding is dependent on what kind of histone codon, or sorry, not codon, histone code has been formed. And that in turn is dependent on the histone modifying enzymes. Now, taking one example, let's say for example, you have a highly compact DNA, and you can see over here that the histone tails have not been modified. So you have the histone tails having a net positive charge because the histone tails have a lot of lysine residue, and the lysine themselves being basic amino acids, you will have a net positive charge. Because there's a net positive charge, it is able to bring in DNA to wrap around the octamers, and therefore you can see that with no acetylation or with no change in the histone tails, you have the DNA wrapped around the histone octamers very closely, and therefore this structure is a highly condensed structure. So if the structure is a highly condensed structure, we know that transcriptionally it is less active. Now suppose you have a histone acetylase coming and attaching a acetyl group to the lysine residues in the histone tail. So then the histone acetylase is considered to be a writer. Why is it considered to be a writer? Because it is making or writing a histone code. That histone code can be read by different complexes. So those different complexes are called as readers. And these readers can actually either make the, so in this case, as you can see, the acetyl group has shown how there is less condensation. So it becomes a less condensed form. So therefore, this is going to be more transcriptionally active than this. Okay, so these histone acetyl transferases, on acetylating the nucleosomes or acetyling the histone tails of the nucleosomes, make the DNA or make the chromatin more accessible to the non-histone proteins. While if you have histone deacetylases, they are going to remove the acetyl, so they are called as erasers. And if they remove the acetyl group, then you can see how the DNA becomes condensed again. So acetylation at specific lysines can form codes that can be bound to by several transcription factors, molecular chaperones, etc. And you can have transcription occurring. While deacetylation erases these codes, thereby leading to less transcription. So this is something that is extremely interesting in terms of chromatin remodeling. Now we go to another group of molecules that help in rearranging the chromatin, and that is something that is called as chromatin remodelers. The word itself is suggesting that these are molecular complexes that can help rearrange the chromatin. So most remodelers are conserved multi-subunit complexes, and they have a highly conserved ATPase subunit. So all chromatin remodelers will have an ATPase subunit definitely present, and they are highly conserved subunits. They disrupt, of course, because ATPase can convert ATP to ADP. The energy that is derived out of it can be used to disrupt the histone DNA contacts and thereby making DNA more accessible. Now, based on the ATPase subunit and apart from the highly conserved region, different structural components enable the chromatin remodelers to be classified into 4 distinct families. And of course, the change in chromatin architecture by the chromatin remodelers is its main or key role. Now, what kind of changes are possible is what we will look at. So, when you have a chromatin and a chromatin remodeler comes and binds, and on binding to the chromatin, the conformational change that happens to the remodeler enables it to have its ATPase getting active, and ATP gets converted to ADP. So, with this, therefore, what happens definitely is that you can have the nucleosome sliding. So, say, consider the nucleosome is assembled in such a way that this gene that has to be transcribed has its promoter region within the nucleosome. But suppose you have this nucleosome sliding, and so this also is sliding, then what can happen is that the core DNA now gets to become the linker DNA, and the linker DNA being more accessible, you can have an RNA pol coming and binding to this region and activating. So this is what is called as nucleosome sliding. So one point is definitely that the remodeler can actually make the DNA compact. That is one thing. The second is that it can change the position of the nucleosome, and therefore that is what is called as nucleosome sliding. Or what it can do is it can remove the nucleosomes completely from the DNA. If it removes the nucleosome core particles completely, you can see how much of the DNA gets exposed, and therefore this exposed DNA is now accessible to all other non-histone proteins. So this is what is called as nucleosome eviction. What is the other thing that can happen is that it does not slide the nucleosome, or it does not change the position of the nucleosome, or it does not evict the nucleosome, but what it does it is unwraps the DNA partially, and on unwrapping the DNA partially, the DNA is very loose around the nucleosome, and therefore the accessibility to non-histone proteins increases. Now, you can see that all this are kinetic changes that are happening to the chromatin, and therefore ATP or the energy is important. You can also have dimer replacement. What do we mean by that? So, for example, you may have an H3H4 being substituted by, say, SEN-PA and H4. Or say, for example, H2AZ, which is a variant of H3 and H4. So, therefore, you may have removal of canonical dimers from the nucleosome core particle, and instead of the canonical dimers you may have a histone variant being substituted. Now that histone variant that has been added to the nucleosome core particle may not have the same kind of affinity to bind to the DNA, and so therefore the DNA that is wrapped around such variants, okay, are going to be loose, and therefore there the accessibility of the DNA to the non-histone proteins or to other molecular machineries become greater. Then one of course also sees that you can have a partial disassembly of the nucleosome core particle. So say for example, there is just removal of H2A, H2B. In that case, what is going to happen is H3H4 will remain bound over there, but the part that is not bound to the H3H4 and is wound but free can be bound to by other accessible or other other protein molecules. So these are the different ways by which, the chromatin architecture is changed by chromatin remodelers. Chromatin remodelers, like we mentioned, depending on the ATPase subunit, the chain differences in the ATPase subunit are classified into four families. So the four families are SWI SNF, SWI SNF family, ISWI family, NURD Mi2CHT family, and INO80 family. These names that are coming, they come because these molecules or these ATPases have been identified or they have been discovered by studying a certain pathway or studying a certain mutation. So thereby the pathway that was being studied or the mutation that was being studied, based on that you have the names being given. Now you can look over here that in all the four families, the ATPase region, okay, is highly conserved. It is highly conserved, and you can see that certain amino acid residues are very highly conserved. So for example, you have D and E over here, right? Aspartic acid and glutamic acid over here, which is highly conserved, and you can see that in all the four families. Similarly, you have the histidine, glutamic acid, leucine, isoleucine, and cysteine being present. These are, you can see, they are present in all the four families. So this portion that you see is highly conserved. Of course, the short insertions or the long insertion, the size of the insertions can vary. So that is how they are classified into different fold. But other than that, there are other DNA elements in the ATPase that have domains that are different from each other. So say for example the SWI/SNF family that has an HSA. This HSA domain is able to bind to actin, and it has a bromo domain, and this bromo domain is able to bind specifically to acetylated lysines of histones. So just in the previous slide we mentioned that histone codes can be read by various molecular machineries, and one of the molecular machineries is chromatin remodelers, and you can already see that the ATPase subunit of the chromatin remodelers can read a histone code. So they can bind to acetylated lysines of histones. So this is one example that very clearly shows that chromatin remodelers do come and bind to the nucleosome core particle depending on the histone code on the nucleosome histone tails. ISWI family has the SANT and the SLIDE domains, which are again histone binding domains. You have the Mi2 family, which is able to bind to histone through the chromo domain, and the INO80 family has again an actin binding domain along with the ATPase domain. So structurally they just differ in certain other domains other than the main domain which is responsible for the ATPase activity. We will look at SWI SNF in a little detail. The SWI basically stands for mating type switching, and SNF stands for sugar non-fermenting pathway. So in fact, it was interesting to note that there were two different groups or groups of scientists who were working on two different pathways that were carried out by yeast, and they were looking at several different mutation based studies. So in vitro mutagenesis leading to them being able to find out the functions of the particular genes that have been mutated. So one of the group was looking at mating type switching in yeast, and the other one was looking at the sugar non-fermenting pathway adopted by the yeast. And interestingly, what was observed is that both were related to ATPase activity. So from that, they came to know that there is something that is influencing these pathways. And then eventually it was found that it is nothing but a chromatin remodeler that is influencing the transcription of the particular genes, and therefore the effect on the particular pathways. This is a diagram showing a analog, or basically BAF is a BRD1 attaching associated factor. This is the SNF that is present in humans, so it's the human homologue. And this is PBF. So this is again another component that is equivalent to SNF in the yeast. And both of these have the ability, so they are basically this belong to the SNF family itself. They have the ability to come and bind to a core particle with a core DNA, and you can see how a loop is being propagated around that. And this loop propagation can lead to two things. One is either sliding of the nucleosome, so this DNA becomes more accessible, or eviction of a nucleosome. So again, you can see that the DNA becomes more accessible. So this is in lieu of which, SWI-SNF acts as a chromatin remodeler, and both the sliding of the nucleosome and the eviction of the nucleosome requires ATP, which is available because of the ATPase subunit of the SNF. And you can already see over here that it has many subunits. So it is a multi-subunit complex. Now, looking at SWI-SNF and how it carries out nucleosome sliding, say for example, what you see over here is that the SWI-SNF binds over here, and after binding over here, because of ATP energy production, you can see how the DNA is loosened. So what happens is that the DNA nucleosome contacts are being broken because of the ATP generated, and you can see how you have the DNA being pushed from here so that a loop is being formed. Now this loop that is formed over here in a wave-like motion, from here it will move to here. Can you see this? See, you have two rounds practically. So the first round looped out, then you have the second round looped out, and this looped out portion eventually comes out. So basically what we are looking at is, if you have a promoter region over here, okay, or if you have a region inaccessible over here, when you have the looping of the DNA and the loop waves out, then the promoter region may be present eventually somewhere here, so it becomes more accessible to the non-histone proteins. So this is what is called as the nucleosome sliding, or the way by which you have the contacts between DNA and histone being disrupted, and that disruption of contact can enable the wrapping of the DNA to be changed. So the region that was wrapped around may not be any longer wrapped around, but a region that was not wrapped around can now get wrapped around. Now the chromatin remodeling complex, that is the RSC complex found in the yeast, uh, shows a similar kind of DNA translocation as observed with the SWI SNF complex. So what you can see over here is clearly that when the RSC, so you have the DNA tightly wound around the nuclear core particle. When the RSC comes and binds, okay, uh, what is observed is that this part of the DNA is translocated. So you have a loop formed over here, as you can see. And this translocation requires ATP to be converted to ADP. So the energy that you derive from here enables the DNA to translocate. So here you have undertwisted DNA waves, okay, and that propagates. So you have the wave moving from here to here. So you can see how the loop is actually getting propagated as a wave, and that loop will come out over here from the other direction. So therefore, this is something that is showing how you can have the DNA wrapped around the nucleosome core particle being changed. That means the region that was part of the nucleus core DNA may not remain as part of the core DNA after the translocation of the DNA around the nucleosome core particle. So let us make the conclusions. Chromatin structure makes the DNA compact, and hence for replication or transcription, when accessibility of DNA to non-histone proteins are required, the chromatin has to be modified. Histone modifying enzymes can alter the affinity with which the DNA can wrap around the nucleosome core particles, Addition of histone variants too can change the chromatin architecture. Chromatin remodelers have a conserved ATPase subunit that enables breaking contact of DNA with NCP. They can modify chromatin by changing position of nucleosomes, eviction of nucleosomes, or disassembly of the nucleosome core particle. Studies with SWI, SNF, and RSC have shown how the nucleosome sliding can occur by looping of DNA associated with the NCP. So basically, the DNA is getting translocated. So chromatin remodeling is a phenomenon that can regulate the molecular processes of replication, transcription, DNA repair, etc. by changing the accessibility of DNA. Thank you.